Good afternoon, everyone. Welcome to the Jones Seminar. So let's get going while people settle down, get their coffee, get something to eat. It's a pleasure to welcome Professor Richard Carson here from Yale University. He's a real distinguished guest for us in the biomedical imaging world, and it's a real honor for him to come up, and we're lucky he could do the drive. He is Professor of Biomedical Engineering and Diagnostic Radiology at Yale, as well as Director of the Yale Positron Emission Tomography Center and Director of Graduate Studies in Biomedical Engineering. He started his career at the NIH, or at least part of his career at the NIH from 1983 to 2005, was in the PET program at the National Institute of Health. And then after that, he was recruited to Yale University to direct their PET center. His research uses positron emission tomography in areas of tracer kinetic modeling, PET physics reconstruction, and PET applications. He lists areas of interest as awake non-human primate PET imaging, 4D and 5D image reconstruction for PET, mathematical model development for novel radiopharmaceuticals, imaging of beta cells in the pancreas, neuroinflammation, imaging in a wide area of disorders, and novel preclinical and clinical applications in oncology. And I think from that you can gather that he has his hands in many, many different applications of PET imaging, and as his role of director at the PET Center is really to help facilitate different applications. He's published over 270 papers by my count, and many of them cited hundreds, and he has 3 papers cited over 1,000 times. So that is an incredible feat as a scientist. Several of those sites have been our own because we study his work quite closely. And so it's a real pleasure to have him here. Please welcome Dr. Carson.
Tomo is Latin for slice. And when we have a positron emitted, it will travel a short distance, annihilate with an electron, and produce 2 gamma rays that go off in opposite directions. So I have this very cute little video that Simon Cherry shared with me some time ago. So you can imagine, here's your chance to have a PET scan virtually there. There you are in the scanner. We're going to zoom in. We have a nice syringe full of radioactive molecules, which of course always sparkle. And then when we push the syringe and inject, we're going to fly through in your veins, go around to your body, and the molecules will move around, and eventually, by random chance, will arrive at any particular tissue. We wish they were smart enough to go where we wanted, and they'll hang around there for a while, jiggling and waiting, when suddenly a positron will annihilate, travel, interact with tissue, slow down, change direction, and eventually it will be absorbed, reach an electron, and the 2 gamma rays will go off. And then the scanner will measure those 2 events in coincidence. And then it becomes a simple mathematics problem to solve for where the images go. Something simple as 50 million equations and about 20 million unknowns. No problem at all. Used to be a little bit. Okay. So here's where we start. We start with a cyclotron. We have this cyclotron in our basement made by GE. And the cyclotron is very boring in its normal closed state. If you open it up, this is the guts of the cyclotron, what's called the Ds. We inject a particle, a proton or a deuteron. It will get accelerated around as we zoom up, and eventually we'll hit one of these 6 targets. These are gas targets, and we'll be able to do some nuclear chemistry. So we'll either make one of our 4 primary isotopes, going from O15, N13, carbon-11, or F18. Most of our work is with carbon and fluorine. Fluorine is our luxuriously long half-life of 2 hours, and carbon is 20 minutes. 20 minutes means nobody can be late. You've got to really be organized about what you're doing, and we use that for a really wide variety of molecules. We hand the isotope to our chemists who are making drugs, radioactive drugs, following CGMP, current good manufacturing practices. That is, we're going to be able to make something that is suitable for human injection. And the key thing that makes that work is we're working at microdoses, working with very, very small quantities, and therefore, a lot for us would be 10 micrograms. And so so that issues of impurities are less of an issue, especially when you have to do your quality control in about 20 minutes. Okay? Here are the 3 human-scale PET scanners that we have, that we're using in our facility. Not on. Is that better? Excuse me. Do I have to start again? No. Mostly for the video. Excellent. Okay. So we have these 3 machines, and they're all dedicated to research. We have a brand new time-of-flight PET scanner for whole body. This is called the MCT. We were really unhappy with Siemens when they named this, and they took the PET out of the name. The M stands for molecular. Somehow the marketing guys thought that was a good idea. An older PET-only machine. And then this one, the HRT. This is a high-resolution machine. It gives us about 2 millimeter resolution, which is really good for PET. And that's really helpful when you're submitting a grant to NIH to say you have one of those scanners to be able to do that. So the game plan, again, our cyclotron will make the isotope. Our chemist will turn it into a drug, a radiopharmaceutical. We'll inject it and scan. And at that point, we can acquire data. Now, clinically, we usually just get a single image. When we're doing it in the research environment, we're going to collect data over time. We're going to try to follow the uptake and clearance of the tracer. So we're starting with a 4D data set at least to be able to work with. What do we apply it to? Well, this is the list of disorders that we've been working with at Yale in the 8 or 9 years here. If you'll notice and go through that list, we're pretty brain-centric, a lot of CNS applications, and that's somewhat due to the particular collaborators we have, but more recently expanding now into cancer, into cardiology, etc. So let me proselytize a little. I know you don't have a research PET here, was what makes PET cool and unique. First of all, we're based on classic radio tracer methods. We are working at trace levels, very small quantities. I disappoint all the freshmen when we say we can inject cocaine in you, but you won't get high because we just don't give enough mass. Our isotopes are nice. Fluorine and carbon are relatively easy to add into molecules. Hopefully there's no chemists in the audience because they're never always offended by claiming that any chemistry is easy. We work with the short half-lives, as we said, 2 hours to 2 minutes. That means we could do repeated studies in the same subject in the same day or from one day to the next. We are quantitative, that we can measure the absolute concentrations of that in a very reliable day-to-day fashion. And we're also very translational, that we can use exactly the same tracers in a human scale or preclinically, often at the same day. We do our best when we make something to apply it to both a human and perhaps an animal scan. And then because we're quantitative and because we have a good physiological model, we can apply tracer kinetics and be able to extract physiology from the images. Now, one of the great disappointments I think in my career, although I won't say it's disappointing, is that most of the math I needed to do tracer kinetics, I learned in my freshman year. Because I basically learned how to do first-order linear differential equations. Because working at tracer levels, everything becomes linear. We're not saturating anything, and life becomes easy. That was the good news. The bad news is we're going to have to do that for every voxel with horribly statistically limited data, so we still have plenty of opportunities to be clever with our data. But here's where we're really clever, the tracers. I like this slide because the font is really small. That's the list of compounds we have used at one point or another recently of different kinds of targets. So this is very exciting that you can take one fundamental technology and look at it at a huge range of applications. And I know you can't even read all that list, but the point is, by working with the right kind of group of chemists to both develop compounds that have been developed elsewhere as well as some of your own, you can look at a wide range of physiology. Whether you're looking at glucose metabolism or protein synthesis or particular neurotransmitters or gene expression, etc. Chemistry is the key. Okay? But it still needs some room for the data analysis people to think about. And part of that is when we have images, how do we interpret them? How do we change it? How do we design our experiment? So here's some example images. And for those of you have your choice of color schemes, I'm showing the exact same image vertically in a gray scale, in a hot body scale, or in a color scale. This is an image. It's a transverse slice through the brain. It's kind of straightforward. We have the front and the back, left and right, or backwards. And we're using a tracer called carbon-11. That's, again, the isotope AFM. It's a Prozac analog. It goes to the serotonin transporter. The site of many, many drugs hit this target. So here's what this image looks like in the first 10 minutes post-injection. Here's what the image looks like from 40 to 60 minutes post-injection. Here's what it looks like at 90 to 120 minutes post-injection. Now, if you're going to use a tracer clinically, you're not going to keep a patient in the scanner for 2 hours. You're just not going to do it. You're going to pick one point. But when you're in the research environment, you have the opportunity to collect all that data, to understand and characterize that, to recognize that the early data is driven a lot by blood flow. In fact, most tracers in the brain look like this early on. The high uptake here is in the gray matter, where there's high flow. The lower uptake in the white matter, where there's lower flow. But progressively with time, the tracer accumulates, or rather doesn't clear from, those regions which have a lot of the target protein this is aimed for. In this case, the serotonin transporter. And here we see that beautifully in the raphe nucleus. Very small nuclei in your midbrain. The source of a lot of the serotonin that goes throughout your brain. So you can have your choice. You could just use these images as they stand and ask questions about differences in disease or effects of drugs, etc. Or you can do your homework, do your engineering, develop that, to develop the appropriate model, to analyze the time activity curve, the images you get for every single voxel in the image. Usually you have to have some idea of the input, that is the delivery of the tracer, because that can change from one person to another and one disease state to another, and analyze it where you take these radioactivity images and produce images where you've separated out the delivery, the blood flow, from the receptor binding. So if I go right back to those images, here's the same data set where now we've processed it. These are the raw images out of the scanner. These are calculated image. Every pixel fitted to a model, extracting the parameters, the delivery, K1 terms, and VT, the volume of distribution. So this allows us to add a lot of rigor to what we're doing in terms of that quantification. We're going to understand if we see local changes, how much of that is due to flow changes, how much of that is due to the receptor changes, to whatever the particular etiology, the particular physiology that we're looking at. Now, I mentioned that we can do this translationally, and we can also do this as mentioned in small animals. So here's some example images where we can be looking at anything, tumors in a mouse, or we can be looking at brain images in a monkey, or in the case here, we're actually being able to look at in a chair mechanism where we can study those awake monkeys. This is a project that we took on a few years ago. It might be the stupidest thing I've ever tried, because not only is studying a monkey's awake dumb, but we weren't even smart enough to lock their heads in. We're trying to let them move their heads. We'll come back to that because it's technology we developed for human primates that we're trying to back translate to non-human ones. Now, animals are really important in what we do in a lot of different areas. This is always that very cute picture. We'll come back to that one. We start with animals, of course, when we're doing with the new tracer. How does it work? Does it hit the target? Does it have the right engagement? How do we interpret it? We do that when we want to validate, because we can do obviously more invasive experiments, give drugs to block that we're not allowed to give in humans. And then even better when we have good animal models of disease. Now, there's a lot of diseases we don't have good animal models. It's not a very good schizophrenic rat model. But there's a lot of good tumor models, although even those have great limitations in many cases. I mean, this depression model in rats with this force swim thing, and maybe it's interesting, but it's a bit of a challenge. This particular one's interesting. That rat is wearing a PET scanner. That's a device called the Rat Cap. It was developed at Brookhaven National Labs. We're collaborating with them. We have that now at our facility. It's actually something that you mount on the rat's head. You have it cantilevered so that the rat doesn't have a heavy head, and you can study the rats awake. We're just getting some experience with learning how to do that. Very disappointing. That image, that rat is dead. Very, very sad. It looks very, very cute in pose, but apparently you can tell that dirty little secret of that one there. So that's not what they look like normally there. But when we do translate, here's examples where you could use the same tracer across species. A tracer of the glutamate receptors, the metabotropic mGlu-R5, where we can study that in humans, and that's a PET image. It's really good quality. We can study in the rhesus monkey, or we can study it here in the rat. Now this is where it's disappointing that the laws of physics do not scale with species, because the image resolution in the rat is pretty poor. And simply because of that positron, that part of that distance, that we have a fundamental resolution limitation in what we can do. But that doesn't mean these data are useless, especially for those of us who've been in this field a very long time. Because we remember when FDG images looked like that on the left. That's what an image looked like back in 1980. I have a film of my brain in 1980 with FDG. And the film looks a lot like that. I've argued that if I got scanned today, the scanners are better, but probably the brain's worse, so it probably would look about the same. But nevertheless, they look a little bit better today. You have a much sharper, much higher resolution, much higher quantification, much better sampling. The image on the right took 10 minutes. The image on the left took an hour and a half to acquire in 7 slices. So we've come a long way in that one. So let me look at one of the technologies, just from a technology viewpoint before we dive into the biological applications. And that's looking at if we're going to scan people for a long period of time, they're going to move. And if we're going to claim to work at high resolution, we've got to correct for head motion. And it's really, really critical when you have the luxury of a high-resolution scanner. Ours is called the HRT. Now, the technology we've used is nothing new there. It's really in terms of it's a tracking system. It's an infrared tracking system we get from Northern Digital. And where you mount on the person's head a little tool that has been calibrated. You have a device behind it to measure it. And you can track the person's motion continuously, about 20 hertz. And then you can use that to correct the data. Now, there's lots of choices on how to do that, and I'll go through some of the evaluation that we did. But in terms of what that looks like, here's what a typical motion trace might look like. People might be very quiet. There might be step changes when they move. They might be quiet drifts as they relax. And we have different profiles here over a 2-hour period. Here's a person using our stylish swim caps with the tool marked above it. So to do that, there's lots of choices how we do that. We do this in what's called list mode, where we take every event, and basically we're going to correct it based on the motion at that instantaneous motion. So we might have an event. Event and PET is a detector pair. Remember, there's 2 gamma rays. We collect both of them. And basically, we're going to, in software, move the event consistent with how the person moved. Okay, so we're going to do that on every single event of the millions or hundreds of millions of events that we get. And we have to do with that. And there's a lot of little details. A detail in PET is a master's thesis, if not more. So there's at least 5 there on that list there, just to be able to get doing all of your homework well. But just to visualize that a little better, here's a view of a subject here. What I'm showing you is this is the bore of the scanner. This is the axial direction, the in-plane direction. And this is the computer representation of where this tool was mounted on the top of the person's head. This person apparently fell asleep, and in so doing, they bolted up and then fell back. And if you look at the original images uncorrected, you'll notice that they don't come back to the same location. When you correct for that, looking at those 2 images on the bottom, we're seeing that we can do a pretty good job of doing those corrections. So this is something we've used routinely. We've done this in about 1,800 consecutive scans on the HRT. It's just part of our routine operation. But the question is, so we've done a technology that's not commonly used, and the question that we wanted to ask for the field is, did you need to do it that well? Was that important? Did you have to correct on the fly for every single event at 20 hertz? That's a job for a graduate student, okay? So in other words, what you want a graduate student is to go back and say, do you need to do this really, really well? Do you have to have perfect correction for everything all the time at high resolution? So the game here, by a graduate student who just left me last year, moved on to work for General Electric, was how to do this is hard. You know, we have a lot of real data, but we don't have the gold standard of what's right. So we did this with simulation, where we used measurements of motion. We have lots and lots of motion data on many, many subjects. We said, let's simulate a PET study where we know the right answer with the motion, and let's say how we can correct it with that motion or with other methods, the more common methods around. See, does it matter to get an idea? So here's how the simulation would work. You start by creating a brain. We use, again, a template. It's called the AL template, where you define a lot of regions. And in each one of those, we're going to assign kinetics based on a particular tracer. In this case, we're going to do a simple 1-compartment model, tissue going in and out. Okay? We're going to correct the time activity curves that we would have that. Now we're going to add a motion profile. We have lots of these motion profiles, the real ones that we've measured in people. And then we're going to simulate the data consistent with the scanner as if we had done that scan. The good news is we have the gold standard. We know the right answer. Now we're going to take that data and apply it to different motion correction algorithms, where we use the motion data, or we just do it with software registrations, etc. We'll take each of those data sets and process it just like we process them normally, and then we can compare those results to the gold standard answers that we have. Nice, big, messy thing, and you want to do this on a large data set. So one thing we have to do is first pick people, decide who we're going to do. We're going to do the big movers and shakers or the nice, quiet folks. We thought we'd work with the people who move a lot. And in fact, one thing we were particularly wanting to do is, since we're doing this at every event, we wanted to look at people who move a lot within a given time frame. Normally when we reconstruct, you're going to bin up the data. You might have 5-minute scans. If people move a lot between scans, it doesn't matter. You can probably correct it. If they move within a scan, you're going to get a blurry image. So we looked at a number of people. This is a measure of motion by individual frame. And if you look, some people are big movers, some people are occasional big movers. And so we analyzed all that, and we said, well, let's pick the movers, see if this is not important to the people who move a lot, then who cares? So we looked at the total amount of motion that we have, and we sampled from our top 15% of our bigger movers, people who move over there. So on an average motion of between 2 and 8 millimeters within frame, kind of an average weighted motion. And then we did a lot of things to this. We tried a lot of things. First, we did the event-by-event correction, where we corrected it just the way we do. And honestly, we're cheating because we have perfect measurement. We're cheating. We know what the motion is. This should be our best possible result. We also said, well, maybe we don't need to do this event by event. Maybe we should just average the data in each frame. In other words, get me the average position and just use that. Measured motion, but not this event by event. Very time-consuming stuff. Or let's do it the way the rest of the world does it. They collect images and then they register them together. They use software algorithms. They use mutual information and register. They might register the final images, or they might be clever enough to not correct them for attenuation. Attenuation, of course, stops most of our photons. If you move between your transmission or CT scan and the PET scan, those original images are wrong. And so in principle, you should do it without that. We also compared it to no motion correction. That's good. It's always good to have some rotten images to compare to. And the static image, which would be the gold standard. Here's some image examples of what we see. So let's hone in on an example. So here's our true image. That would be perfect. Here's our image that's stationary. So that's what a typical image would look like. It's a little bit noisy, and that's part of it. Noise is a limiting factor. If our improvement is not improving things over the noise, who cares? So here we're looking at that small structure. This is also using AFM, the images I showed you before. We're looking at the RAF-A nucleus. And if you look at the substructures here in the RAF-A, we reconstruct them very nicely with the event by event. If I use the average motion, we're doing okay. And progressively with the other methods, not quite so good. And of course, with no motion, it's a disaster. If you look at numerically how well we do, we compare our values as a function of the magnitude of motion. The red is the event by event. Compared to stationary, we're getting good agreement there. But we're really not hurting too badly. By the standard methods most people use, the registration of the images, they're in the blue and the yellow, except for the people who move a lot, you're really only getting a small percentage of error, which was pretty encouraging, at least for them, that it turns out that it isn't that critical. So did you need to do it the hard way? Well, not really. Except for the real large people who move, maybe the top 15%, and even those are only producing about a 10% bias, 10% error, then it's really not a big problem. This gets interesting when you're doing a clinical study. If your clinical population moves more than your healthies, or vice versa. If you're doing some movement disorders, it could be if you're doing Parkinson's disease, they may be sitting still better than your healthy controls. But we still like the hardware motion for a couple reasons. When you do it in software, you really have to tune this. Every tracer is different, and each image registration requires a lot of details. On the other hand, we've had some challenges with our hardware correction, mostly depending on hairstyle. That we were using this lovely swim cap, and depending on either people who have too high sheen hair or bald's usually okay, but to get that right takes a little bit of care. We've gotten better at it. But probably a better technology, we've begun to work on somewhat of that, is to do something where you don't have to actually connect anything to the head. You can use Microsoft Kinect, or you can use some other camera systems. Lots of challenges with them, but I think that's where we'll need to go. At that point, maybe that'll get incorporated into the scanner. Now, I mentioned that we think we've solved this issue. It's at least in good shape, so then we took on doing awake monkey imaging. And so here what we've done is we have our animals in this chair, and they're trained to be in the chair. And because our scanners can't go all the way down, the animals don't like to lie down, we kind of meet them part way. We tilt the scanner about 45 degrees, and the animal tilts back. We have to train animals to be able to do that. And then the idea is we're going to use that same Vicrit with the monkey's head. And like any good thing, you don't ever show a picture of a monkey in a presentation. So we have our nice furry monkey there, and be able to track that motion along the way. So this took a lot of work. And we found, you know what? Monkeys move faster than humans. Okay? And no matter how well we're feeding them or paying them or whatever. And so Things like, synchronizing the time between the motion acquisition and the computer, you better get it down to about 10 milliseconds to get good accuracy, because the velocities they can get are really incredible, as you might imagine. So this is great. I mean, again, great if you've got grad students to be able to dedicate to this stuff. So we can look at it, and we've done this a couple different ways, and I don't think I need to belabor it. We did something where we first did a method looking just stopping when they moved a lot and being able to do that, or looking at the event by event. And when you look at some of the structures here in the various different ways, we can claim that when we do the event by event, we're doing pretty well. If you look at, I think, this column is an anesthetized study, so that'd be the best we could do, and then we think we're doing pretty well with the event by event. But it's still something that we're not sure this was a good idea, because they move so fast. And it's also issues where you have to do the physics really well, because if starting in the middle of a scanner and I move to the edge, the resolution gets worse, and I have to correct for that very, very well. So this was one of these nice projects that said, well, it's great, and we're not sure how well we're going to do with this, but it did stress all of our technology there. So let me switch from more of an engineering problem to more of the biological or clinical problems, and I want to focus on brain receptors. And just to be sure we're all on the same page, if we're looking at a brain, we're looking at there will be various sites that will have neurons that will be producing various neurotransmitters. And these neurons will project with their axons down to hitting the dendrites of postsynaptic neurons there. And if we zoom down, what we're looking at here, for example, is a dopamine receptor, where we would have a nerve impulse would cause the vesicle to exocytose, would release the neurotransmitter into the synapse, and then would bind to a particular receptor or transporter on the other side. And so now our trick is to make molecules that let us interrogate different aspects of that, binding to the receptors, part of the synthesis pathways, or in the presynaptic sites. So what can we do? We can look at the absolute quantification. How does that change in disease? How does that change under treatment? We can look at occupancy of this, and we'll come back to that, because it's a matter of, because we're working at tracer levels, we aren't affecting the sites. If you block some of the sites, our signal will go down. And that really looks a very cool thing to be able to look at the dynamics of neurotransmitters in live human beings. So like right now, you're all really enjoying this lecture, I hope. And your opiates are just pouring out, and so if we could do a scan with carfentanil now, you'd have a black hole in your brain, because you'd be totally saturated with your endogenous opioids there, at least if you had the caffeinated coffee. Okay. So let's show you some images first. It's always good to show that. Here's an example, and I don't know if we can dim the lights a little more. That might be helpful. This is a target. The tracer is called methylreboxetine. It's specific for the norepinephrine transporter, one of the monoamines in the brain. Has a very specific distribution, primarily here in the thalamus and then down in the midbrain. And if you do this spinning, you can really be able to see some of these very small structures down in the midbrain. We've applied this in a number of different disease states, from cocaine, drug abuse, Tourette's, and depression. And one thing that's very exciting, the source of norepinephrine in the brain is something called the locus coeruleus. It's really small. But when we zoom down and analyze the images and look, although we can't absolutely prove it, we're seeing the site of that in exactly the right position and shape consistent with the locus coeruleus. So either we're being lucky, or we're being good, or probably a little bit of both. This is very hard to do with the kind of resolution that we're talking about. This is not common to look at. These are extremely small structures in the midbrain, and by having the ultra-high resolution scanner, better than what's normally available clinically, we can able to ask this. But it really poses a lot of technical imaging questions. How do I measure this reliably across subjects? Here's a different target. This is the cannabinoid receptor. This is the target site of THC, as in the active ingredient in marijuana. This is one of the highest capacity receptors in your brain, which is really, really quite interesting. Not surprisingly, we're studying cannabis use, but also in schizophrenia. This is a really important target in obesity and a wide variety of disorders. It is ubiquitous throughout the brain. A different one, here's a serotonin ligand. There's lots of different serotonin targets. This is the serotonin 1B. We've been able to study that in also a wide variety of syndromes there. It has some interesting distribution in the occipital lobe and in this area, which is called the globus pallidus. We found some interesting correlations of that in disease. This is an example. I'm showing this one in part because sometimes when you say, is there a difference in this target in this population, there's so much heterogeneity and variability you need a huge cohort to be able to prove it. But what you can find often are interesting correlations. Here versus age of offset, this is as a function of trauma control for a PTSD study. Sometimes it's your behavioral task. I'll show another example in a couple of slides. So it's one of those things where you're not always going to show a disease-specific differences in regions, but how that interacts, how a specific behavioral construct is related to the local concentration of receptors in different areas. Here's another target. It's a dopamine receptor, but this one's a little different. This is called PHNO. It's an agonist, and it lets us see the basal ganglia, or if you look carefully, it looks like some bunny rabbits. They're very cute there. We're also seeing these other structures called the substantia nigra. That's the source of dopamine in the brain, and we can visualize them very, very well. This produced some really interesting relationships when we looked at subjects where we looked both at PET with PHNO, as well as looking at other measures. In this case, this is a collaboration with Mark Potenza, where we're looking at binding of both two different tracers. Or let me start down here. Here where we're doing functional MRI and looking at a task, looking at bold change in different regions, as opposed to this dopaminergic signal in the ventral striatum. And what's really very interesting is how we get differential correlations across subjects. Or even more interesting, this correlation is spectacularly good, is looking at two different tracers in the same subjects. Here we're looking at the dopamine marker, the substantia nigra. It projects up to the frontal cortex. Here we're looking at that with the serotonin 1B. And we've got this spectacular correlation. Now this is one of those things that you look at that and say, oh, you got lucky. And so what do you do? Well, okay, we're going to do two more subjects. And damn, they just laid right on the line. It was one of those really nice things. What does it mean? We're not sure, but it is reflecting part of the pharmacology network that we can add along with the kind of functional connectivity information we can get with fMRI. Here's another one. I show this one in black and white because the image is so good. This is, again, that mGluR5 glutamate receptor. This is an interesting one. Glutamate is your primary excitatory neurotransmitter. And we've studied that in a variety of different disorders that we're beginning to do. But it's one of those where we're very, very interesting to see how this changes in different disease states and also with different activations. We've just published a paper showing ketamine effects with this, suggesting it will be sensitive to glutamate changes, which would be very exciting. And the last one I think I want to show you, again, the serotonin transport. I've showed you this one before. It has really just some beautiful topography where the distribution of the serotonin transports and heading down into the midbrain and then down into the spinal cord. And so this led us to say, well, gee, maybe we could use this to study spinal cord injury. In the spinal cord, there is a lot of serotonin. And it's an old system phylogenetically, and it's one where you can be able to measure. And so we thought it would be an interesting model to try to do this, to look at spinal cord injury and spinal cord regrowth. This is a collaboration with Steve Strittmatter in our medical school. So how do you start doing that? Is this going to be sensitive? Well, you start with a rat. Here's a rat image. And on the image, this is a cross-sectional image of the rat. You see the rat brain. We've hypersaturated the image. And there's the spinal cord. And we're seeing this beautiful spinal cord. We're seeing the serotonin fibers in the spinal cord. Hopefully you're thinking, damn, they're small, aren't they? And they are. The goodness. So how are we seeing this with that rotten PET resolution of 2 or 3 millimeters? The trick is we've got great contrast. You're seeing a poor rendition of great contrast, which still comes out as very good contrast. So we might be at 10 to 1. We're probably 3 to 1 here, but we can still see it. Then we said, well, that's all well and good. Is it a specific binding? So you give a drug. You give citalopram, an SSRI, and it's gone, and you wipe it out in the same animal. You do that on the same day in the same animal. So you have a specific ligand. Now let's take it to an animal model where we've lesioned it. And now when we lesion the animal, you're able to show that you lose the serotonergic fibers beyond it. And then when you treat those animals, and this is a paper we published a couple years ago, it looks like we're getting increased uptake further down the pike. And I don't have the good images to show you. So what do? Of course, you write a grant and you go into human beings and you're sure it's going to work, and it didn't work at all. And the images just were terrible. We don't see that. We don't see the specification. We did better measuring this in the rat with a very small spinal cord than we did in the human being. And we've looked and we've tried to understand why, and it's not an imaging situation. It just may be that the concentration of the site just isn't high enough for us to give us the good contrast. So that's the trick. When you pick a compound, how do you pick a compound that's going to work? And this is where there's serious magic. How do you develop a tracer to be able to be usefully to measure? I've shown you a lot of tracers, and some of them are good and some of them are great. But, it's a complicated area. Now, people have been developing drugs for years, and they have a pretty good understanding of what it takes to make a good drug. So if you're trying to hit a target in the brain, you need a good affinity. That's called the Ki. You need it to be selectivity. Well, you want it to be able to hit your target. It's not too critical. You want it to be pretty lipophilic, so it's going to get into the brain and hang around a long time. And you'd like it to have a long, slow metabolism. When we do tracers, it's different. We need to have really, really high affinity. So we have very good binding at very, very low target concentrations. We need it to be selective. We want it to only go to one site, otherwise the image is going to be a mess. We're going to have a compound of different sites we're seeing. We want it to be lipophilic, but not too lipophilic, just enough that it's going to cross the blood-brain barrier, but not stick to everything. And we don't want it to hang around too long. We need it to clear to come to equilibrium. So those images you saw, so the background would clear out. Now, in real drugs, you're really worried about toxicology because you're giving good mass doses. In PET tracers, we don't really care because we're giving microdoses. Some of the very good PET tracers for the nicotinic receptors are a poison at pharmacological doses, but they're fine if you do your homework carefully. So the space of chemistry that we have to aim for gets very tight. There's a long list of these different kind of things to balance that we want to do, but let me just focus on a couple things, is looking at something simple, affinity. Affinity says how tightly does the molecule bind? How well does it bind? And this is where we have some trade-offs, that if we have low affinity, it means we're not going to bind very much, we're not going to have a lot of signal. We're not going to have good signal to noise there. But too much affinity is bad too, because what that usually makes is the kinetics are slow. Now, if you think of it, first the tracer's got to get to the brain and get in. Now, if I have a really high affinity compound that as soon as it hits a receptor, it sticks, well, that means every molecule that goes in is going to stick, which means if I have twice as much or half as much receptor, the signal is identical. So too much is not a good thing. Now, the other part becomes, the other alternative is that lipophilicity. We want it to be lipophilic enough to cross, so to be able to zoom through the endothelial cells into the brain, but we don't want it to be binding to too many things. So again, we need to find these Goldilocks tracers that have just the right amount of lipophilicity and affinity, and not too many of these other factors. So let me talk about an example of something we've developed and been publishing lately of a target for the Kappa opioid receptor. Opioid receptors, I've mentioned those before. There's primarily three different kinds, the mu, the Kappa, and the delta. Opioid receptors were some of the first receptors that were ever really developed, found, and sequenced back in the 70s, and that's why they aren't numbered. That's why they got Greek letters. All the other ones get numbered because there's too many of them. And so this is this compound. You'll notice those letters LY. That stands for Eli Lilly because we collaborated with them in development. This was a drug. This is the number in their catalog. It's a very catchy name, 27950. We really hate these numbers. It's for people who like numbers, having these long numbers get very, very tedious there. In vitro, this looked good. That meant when we take out brain tissues, we said it was very selective. We look at the affinity for the Kappa compared to the others. I've got like this 60-fold selectivity. That's good. I'm probably going to get a pretty selective measure. So what do we have to do to be able to do this? We've got to figure out a radio labeling scheme. And so this is an example. This one's pretty complicated. So this is what we worked out. You have to start with what the cyclotron can produce. It produces carbon dioxide labeled with CO2. We turn it into methane. In this case, we turn it into cyanide. And then we have a precursor, which we're going to add and then have to formulate to make remove a protective group, and then we can get to our final compound. This is a complicated synthesis with that many steps for carbon-11. When we have that along, we might take that into monkeys. So here's some views of a monkey. These are different directions of a monkey scan. To show, is this specific and selective, we'll give blocking agents. We'll start with naloxone. That's a general both blocks mu and kappa, blocked everything. We have a selective kappa agent, so we're pretty comfortable that we're barking up the right tree there. And then we can take that into humans. So here's an example with the human where I'm not just showing you the raw images, I'm showing you those processed images. In this case, a binding potential image. We've done the modeling pixel by pixel. And we see this just gorgeous uptake here in the amygdala, seeing it in the insula, and a bit in the cingulate cortex there, places that are known to have a lot of the kappa opioid receptors. Now we have a tool that we can use for a variety of different syndromes. We can also look, in this case we're studying this, for example, in alcohol syndrome, where naltrexone, which is a kappa antagonist, is being used pretty routinely to help treat this. And we're going to be able to learn how much does naltrexone target those agents, and is that different in the people who successfully wean off of alcohol compared to those controlled. So we can find that a clinically relevant dose of 50 milligram blocks about 80% of that target. And we'll be able to titrate that to figure out what level that we're looking to need. So this is part of the story. To build a tracer that gives you a specific signal, you can use it to interrogate disease, but it's only part of the story. And part of the reason is that receptors, many of our receptors, so-called G-protein couple receptors, exist in two forms. They can exist coupled to G proteins in what's called their high affinity state, where they bind effectively to the endogenous compound, or in their low affinity state, in which case where they're not so specific. When the pharmaceutical company works towards that, you're producing a drug that either turns that on to an agonist that hits only in the G-protein coupled state, or an antagonist, mostly what drugs are, which binds on either state indiscriminately. You can just think about it as a different key, whether it's a master key or a different key that fits into the lock but may not turn it. So what's very exciting that we're in the midst of now is being able to use two different compounds hitting the same site, but one seeing only the G-protein coupled site that is in its high affinity state, and one is seeing it in both states, agonist and antagonist sites. So I described the antagonist tracer we developed with Lilly. Here's another older compound there that we tried to do for an agonist. Now, an agonist means if I give it, it's going to hit, it's going to have a pharmacological effect. So I've got to be really, really careful, know at least 2 or 3 more reallys, that we're being sure that the mass we give, the micrograms we give, are well below what's going to cause that pharmacological effect. Now, in the world of PET, that becomes the word specific activity. Okay, telling me how much mass. Specific activity is a ratio. It's a ratio of the number of hot molecules to the number of cold molecules. High specific activity means I'm giving plenty of hot and very, very little cold. Here's my quiz for the grad students. So if I have a chemist who has a really, really good day, and he makes a batch of compound about what fraction of the molecules he makes are radioactive. So half of them radioactive, 90% of them radioactive, 10%. If I have a great chemist who has a really good day, he gets 1 out of 1,000. That's a really good day. So, in fact, for every hot molecule, we're sending about 999 more cold molecules along. So we've got to be really, really careful to do that well. I've often given a great shout out to Avogadro for thinking very big when he chose that number of molecules in a mole so that we still have enough counts to be able to measure and still give a very small fraction of concentration. So to develop this particular tracer, the standard method gave a pretty poor specific activity, about 1, and this was actually a dangerous level in terms of getting enough counts in terms of the mass we gave. And so we came up with a new synthesis with about a 9-fold improvement, and that let us move on to humans. So we're collecting that data now, and forgive me for the different color scales, but being able to, in the same subjects, looking at the agonist and antagonist targets and to see how they differ and how that changes. Because what it might be in disease is the number of receptors may not different, but how many of them are in the high affinity state or how those cycle in disease states, and to be able to elucidate that in live human beings in disease is just a very exciting opportunity that we don't think you can do with any other technology. So we've talked about the receptors and the targets, but we're not just sensitive to the number of receptors, we're sensitive to how many of them are available. So here's this simple cartoon where we can think about availability, occupancy of receptors. So we've zoomed down into our cartoon synapse, and we have our dopamine D2 receptors there, and we have our dopamine molecules in white, and they're hanging around the synapse, and some of them are bound to the receptor. And say we give our radioactive molecules are always red, and that one's called carbon-11 raclopride, and that will only be able to bind to the available receptors. And of course, this cartoon is silly because we won't be saturating all of them. Now, if we change the dopamine, like we talked about before, we're going to increase the dopamine. Here we're giving amphetamine. It's going to pump the dopamine out. We're going to have a lot more dopamine. We're going to see less of our tracer. So that's very exciting that we can not just look at the hardware, that is the protein, the availability of that, but the dynamics of that, how that's changing, whether we're doing that with your endogenous neurotransmitters or with an exogenous drugs. The pharmaceutical companies have figured this out. And they've gotten excited, at least some of them, about PET. So what do they do is they would like, if they have a particular drug targeted at some receptor, and they would like to know at a given dose how much occupancy are they hitting, what do we need? We need a PET tracer that hits the same target. And then we do a design that looks like this. We would take a subject, and we often can do this study about 9 subjects. We'd get a baseline scan. We'd see how much available receptor we have. And then what we would do is give them a dose of some tracer. So we do a bolus injection. We collect the scan and do all that modeling stuff that we do. And then we would compare after you give the drug to before you gave the drug. And what's cool with this carbon-11 is we could actually get 2 scans post-injection because the tracer goes away. Now, the first thing that we'll get out of here is images. This is the slide I call the vice president slide. These are good pictures. Every vice president of drug company can relate to this picture when we say, see, that's what your drug did between the first scan and the second, and you blocked all those sites. We turn that bright yellow image into purple. And then, even better, when we bring that subject back the next day, you'll notice that the availability of the receptor is coming back as the drug clears out. That says they've engaged the target. So these are the pretty pictures that you want to give that people get very excited about that says their target. Particularly, this is the histamine H3 receptor. Now, that's good for sales, but for science, you've got to do your homework. You've got to do you've got to look at your time activity curves, you find your regions of interest, you're going to analyze them with models. I don't need to go through all that, come up with the numbers called the binding potentials, go through your calculations to get numbers out of this. And the real money slide on this is this one. So let me walk through what we have in the data set. This is about a $600,000 slide, because that's what one of these studies will cost to do. This is done in 9 subjects. It's done in the process of about 3 months. So the solid symbols refer to the occupancy of the person at the maximum concentration in the blood. That's this scan compared to the baseline. The same person came back at 24 hours, their drug levels dropped, and now they have lower occupancy. They are showing some of the binding returning. And this is very simply looking at a ratio, a quantitative ratio of the availability from here to here. Now, what we're comparing that to is the drug level. There we're going to take blood samples, measure the pharmacokinetics, the plasma kinetics of that, and compare it. That solid line is the classic single receptor site relationship between them. And so you absolutely know for that drug exactly what concentration will produce whatever level of occupancy. We're still arguing about that point, who screwed that one up. We think they screwed up the PK, but that's still up for grabs. But this is a pretty good data set. You're talking about doing this in a very small number of subjects in a very early phase trial. This is done almost immediately after you've done the safety trial of the drug to be able to do that. Now, when I show that slide to freshman biomedical engineers, or freshman people taking a biomedical engineering class, I ask them, now, why is it that a highfalutin Ivy League university like Yale would lower itself to helping a drug company to develop their drugs? And they all raise their hands and say, money! And that's good. That means they know that. But the sweet spot here is when, by working with the companies who have these huge libraries of compounds, they give us good leads for compounds that will become good tracers, like you saw with the Lilly case. And that's when we get the win-win scenario about not only do we answer their question about that drug, but now we have a new tool for clinical research, a new tool for clinical evaluation that we can take. And damn, it helps those grant applications when you've got something that's never been done before. So that's usually a very, very good thing. All right, so let me change gears a little bit. Let me drop below the neck and drop down to the pancreas. Okay? This is interesting how we got to the pancreas. But the pancreas and the brain have a lot of things in common. So the pancreas, we're talking particularly about the beta cells in the pancreas and the Olives of Langerhans that secrete insulin. And it's a critical organ, obviously, to look at in diabetes. In type 1 diabetes, we lose the beta cells completely. In type 2, you become insulin resistant, and there's a time-varying change in the release of insulin and time effects for a while. In fact, there's upregulation of the beta cells to produce more insulin in an attempt to deal with the lack of insulin sensitivity in the body. Now, the tracer we're going to use to look at this was developed for the brain. It was developed for Parkinson's. And the particular one is a tracer that goes to the vesicular monoamine transporter. We'll come back to that in a second. So first, let's go back to the pancreas. What our goal would be is, can we measure the mass of the beta cells, those cells that produce the insulin? Because we want to be able to follow that in disease. We want to follow that under treatment. We understand the natural etiology of that in type 2 diabetes, etc. So first, let's do a little bit of the physics there. First of all, the pancreas, unlike the brain, nice, hard, strong, big organ. The pancreas is kind of this little feathery thing that goes around here. And inside of it, there's only about 1 or 3%, that range, are the beta cells. So we're already trying to say, well, gee, that might be a difficult imaging target. If you only have the specific target you're looking for is only 1 to 3% of the volume. Of course, in the brain, those dopamine receptors, they're even less. So as long as we have something selective enough or specific enough, think about that rat spinal cord, we're still going to be able to image it. All right, so now what's the mechanism we're using? So this mechanism is this vesicular monoamine transporter. Monoamine is dopamine, serotonin, or epinephrine, one of those neurotransmitters. And first, let's look in the brain. What happens? There are these specific proteins, the VMATs, that transport the monoamines into the vesicles in anticipation of releasing them during an action potential. Interestingly, or serendipitously, or by design, those same kind of vesicles, those same proteins exist in the pancreas, where there's a insulin and where dopamine is co-released with insulin, and therefore there's a VMAT2 target there. So the hypothesis was that VMAT2 would be a good target. It would be a good protein target that would be indicative of beta cells. Not exactly beta cells, but something there that's relevant. So you go back, you do your immunohistochemistry. This was work done by others, be able to look at VMAT2 expression and looking at insulin expression, and their co-expression is excellent in the pancreas there. And now you need a tracer for that. And now this is where it got interesting, because the same target was being used in the brain to look at the presence of vesicles, that is the presence of presynaptic neurons, something which is tremendously reduced in Parkinson's disease. There were 2 tracers that were developed, dihydrotetrabenazine, DTBZ, and another analog of that, fluoropropyl. The dihydro is labeled with carbon-11, that's that 20-minute half-life. This one is a fluorinated version, so it's a slightly different molecule, an analog. It's got that 2-hour half-life, so it has a little bit better statistics out later. And both of these compounds had good affinity. The affinity number is lower. That means higher affinity for the fluoropropyl DTBZ. Okay, so let's try it. Let's see if this molecule is going to be useful to measure beta cells. Where do we start? We're going to start in the rat. Okay, so we're going to do that same kind of thing. We want to say, okay, is there specific binding? Can we measure specific binding in the pancreas of the rat? Can we demonstrate that in vivo? Well, here's a nice thing. We can do that even within one study. So here's a side view of the rat. There's the brain. There's uptake of this tracer in the brain. I think those are salivary glands you see in the liver. And nicely, the hottest spot there, that red spot, is the pancreas. A lot of uptake here. That's great. So here what we're measuring is the time activity curve. We've injected this rat. We're following it in time. We're reconstructing images. These are those values in a normalized unit called SUV. So blue is the kidney. We're showing some clearance. Green is the liver. Red is the pancreas. Now what do we do in the middle of the study? We give an injection of just the cold compound. In other words, we're going to give a cold amount just to block it, just to displace it. And that's where we get this lovely immediate displacement of the tracer there. So we're seeing live in one study the fraction of the binding that we can attribute specifically. That's a great luxury, being able to do those dynamic scans and being able to follow things in time, not having to sacrifice animals or do that. We can do that at the same time. We can, of course, sacrifice the animals as well. Okay, so that says something that says that, well, at least some of the binding is specific. We can quantify that, and we can be able to look at that as a function of that. So we've looked at that magnitude, and we found, okay, that we think that of that signal, about 1/3 is non-specific, non-displaceable, 2/3 is specific. So now we want to take that into an animal model, and we're using an animal streptozotocin model where we've killed off the beta cells. What we had hoped for was that we wouldn't see any specific binding if all the VMAT2 was in the beta cells. In other words, we'd measure the blue. No such luck. There's still some specific binding. We still see some of it. That says that some of this target is not in the beta cells. Okay? All right, so we've got one strike against this tracer already. We don't have we have some background that's non-specific. We also seem to have some specific binding that's not associated with beta cells. So now you have to make the call. Is this good enough to go to human? Yeah, we went anyway. We went on to human. Okay? So we did a study in type 1 diabetes. So this was kind of meant to be, well, if it doesn't work here, in a population with long type 1 diabetes, 19 years, long time, then it's very unlikely. They should have no beta cells. They've got no beta cell function left. If we can't see a difference here, this isn't going to work. Well, we check the subjects first. We do some arginine stimulus test, measuring their glucose release, and we could see that in healthy controls, we get a normal insulin release. In these type 1s, we're getting nothing. So that's all fine. Now we're ready to try to do the imaging. Okay? So here's what the image looks like in a human being. What you're showing is 3 views here. We're seeing a sagittal view, a side view. We're seeing a coronal view, and then the axial transverse views. And nicely, the red area, again, is the pancreas. This is one where the rat results and the human results came out very, very similar in terms of uptake and distribution. We don't always get that. You're seeing, in terms of the topology, you're seeing liver uptake here. We're seeing spleen there. You see a little bit of kidneys and a little bit of heart. This is the bone marrow. Bone marrow tends to accumulate a lot of what we do. It's not the spinal cord. Otherwise, it would be a continuous readout there. So that was a healthy control. We then did it. And what I'm showing you here is the first 4 healthy controls we saw and the first 4 diabetics that we saw. And this is very exciting when the image tells something, and these are all on an absolute scale of radioactivity so far, that we got nice 4 red pancreas in the healthies, and they're there. They're not gone. They're more in yellow, etc. But we do see a significant reduction. And then when we take and do the math, now we have some choices. We can just look at the images. Just look at a late image, something called a standard uptake value, and we got a nice statistically significant reduction. And we got it in different parts of the pancreas. But then we said, no, we're engineers. We do this right. We're going to analyze that. We modeled it and went back and forth and came up with something called that volume of distribution. You saw that before. And there's still a reduction, but it stopped becoming statistically significant. So we tried another measure, the so-called binding potential, where we normalized it. Now that's a little tricky, because we're jumping to a conclusion. Here we use kidney. We're hoping the kidney is not different. Now, numerically, they're not different, but maybe it's not. And there we got our statistical significance back. But this is kind of tricky. These studies take a couple hours, long time, blood samples. This is just an image. And the question is, well, this showed a better picture. Is it showing us the right answer? Well, we'll come back to that. Okay? We did a number of other comparisons. We looked at how our measures correlate with C-peptide release, a measure of insulin function. We were really happy that the one person who seemed to show the lowest value seemed to have the lowest peptide release. Maybe this was really real. And then we also recognized that one of the challenges we have is that the pancreas is actually smaller in type 1 diabetes. And we have to be careful about that. If you remember, we have only so much resolution. If I have two identical objects and one smaller, if I'm not careful, I'm going to have a lower value, partial volume effect. We think we did it carefully, but when we correct for that, it's even a bigger effect. Depends on if you want to use that as an advantage or a disadvantage. So what we published was, it worked. We said, you see, we can use this to image the beta cells. Now, it's far from perfect. It's not like we are, because this, again, is type 1 diabetes. There should be none of this at all. And we reduced things. We reduced the total binding, but not dramatically. In fact, if you go back, and let me come back to these, you'll notice that the reduction in the binding we have is very similar to what we saw in the streptozoosin rats, that we dropped by about a third. Okay, that there's still some non-specific, as well as probably some specific elsewhere. Now, we can't give these big blocking drugs to the humans because we'd probably kill them. That would probably not be a good plan. So we have a lot to do, though. So this looks interesting, but we still have a lot of homework to do. And this is where we are right now. How do we take an interesting tool and say, what can we get out of it? How can we tailor? How can we think about all of the issues that could be confounding what we're doing or making it better? We'll see. So I'm just going to highlight a few of these in the closing minutes here just to get a feel for the other things you want to think about. Okay, well, the first thing we want to think about is when you inject something in the body, your body has this nasty habit of chewing it up, of metabolizing it. It's true with almost all the molecules that we do. And our scanners are good, but all they're measuring is the isotope. We don't know what they're bound to. Now, the brain is very helpful. It has the blood-brain barrier. And if the metabolites don't cross very well, we can hopefully find that what we're measuring is the good stuff. There is no blood-pancreas barrier. And so a worry we had is, well, maybe part of what we're seeing are metabolite. So how do we look at that? Well, there we need an animal. In this case, we were doing this in a baboon. We had a baboon reaching end of life. We injected them with the tracer. We took out the pancreas. There's a nice piece of pancreas meat. I don't know what the right recipe for pancreas is, but whatever it is, it's not good. And we were able to do the metabolite measures, looking using high-pressure liquid chromatography, HPLC, and being able to look at that. And although this slide's a little bit hard to see, we had lots of metabolites in the blood. So this is the blood profile in the red. In fact, at the time of that scan, a huge fraction, probably over 90%, has been metabolized in the blood. But if we look in the pancreas, in the red, there's a very, very small amount of pancreas. So this would be our percentage in this case, and we were happy or lucky that there's very little metabolites at all in the pancreas. It's mostly the tracer. Now, if you remember a few minutes ago, I said we normalize it to the kidney. Well, the kidney's not so good. There's a lot lower activity in the kidney. It's showing some metabolites. Some of them are getting in. They may be accumulating in there. We have yet to decide. So now we have a challenge. We say, well, the pancreas looks good, but we don't know if we want to normalize it to something. We'd like an organ that is not sensitive to metabolites. Now, why does that matter? Well, it may not matter. If all my patients metabolize in exactly the same way, maybe that'll be a consistent error. We like consistent errors. We make them all the time. But the trick is, do we know if they're there or not? So we thought, is there a better way to say? Can we find a way to say, what's the good background? How can we do in a human being and say, what's the background? What's the non-specific binding? We can't give a drug to block this. We don't have one that would be safe, especially because it goes to the brain. So how can I make a molecule that does everything the same as my tracer, but doesn't bind specifically? We go to chemistry. We use an enantiomer. Okay, so the particular molecule, the plus version, is the one that specifically binds. There's a minus version. There's a mirror image version. We can label that too. And the affinity for that is thousands of fold worse. So that was that logic. Can we flip to an affinity to be able to do that? We started that in baboon. So this is what the minus scan looks like. And this is the time course. And we're seeing nothing like what we see with the hot scans in the plus. And then we did some displacement studies like we did in the rat. And that we could see that if I give cold plus, that is non-radioactive, I can kick it off. If I'm giving the minus, it's having no effect. Okay? So that sounded like, well, this might be a good tool. And we've begun to do human studies. But then we thought, well, maybe we better check about how do we interpret the minus data. And we went back and did some of those baboon studies to say, is the minus measurement also pure compound? And it is for the plus, but it's not for the minus. So to put that together, sorry if that's fast. So we wanted to get a measure of the background, the non-specific. We said, what's the best way to do that? Use the mirror image. It's going to clear the same. It's going to have the same lipophilicity. And the problem is that it seems to show uptake of metabolites. And you're saying, why is that behaving differently? It really isn't. It's just that this is a problem that there's so much less uptake that a very small amount of metabolite, maybe 3% here, translates into 45% there. And it's a huge background. So we're kind of stuck. This is where we do appreciate the brain. Brain does a lot of nice things for us. Not only does the brain avoid metabolites, but it doesn't beat and it doesn't breathe, because when you breathe, things move. This is another problem we're dealing with when we go below the neck. This is a gated image. So we've taken a 10-minute image. We're taking one cross-section. In this image, we're seeing the pancreas. This is an early image, so you see the kidneys and the liver, etc. And we're just showing you images where we're collecting it, where we've taken the data at each point and just breaking it into different bins and showing that as an image. Well, we should be able to handle that. We did that motion correction before, and we've started to use some of the same technology to do motion correction now in the body. This is big and messy. We've got to kind of calibrate. We measure a respiratory signal with something called an enzyme belt that tracks that continuously over time. We have to kind of calibrate that. This is very hard to see, I apologize, where we analyze those gated images and try to calibrate the position of the pancreas with the average position of the motion. Maybe that's going to be more visible here. So we've taken these gated images, it's a function of the respiratory gate, segmented out to try to find where, in this case, we're seeing both the kidney and the pancreas are. And then we want to calibrate that to the signal that we can measure continuously in the person. Okay? So that's what this data is. We're showing the continuous correlation between the displacement of this external trace, this enzyme device, to the center of mass here of the pancreas and the kidney. And we were very happy because we show very similar relationships. In other words, in this part, this field of view, everything seems to be moving together. There's a lot of corrections to this in place now in commercial scanners. Mostly they're handling the Z motion, the up and down motion. We're also finding some significant front anterior posterior motion that we're able to correct. And I can show you what those images look like. So if we can look there, I'm showing you on the left. This would be the blurred image without correction. This would be a gated image where we're only using it at one part of the respiratory cycle. This means we've kind of frozen motion, but at the same time, we have much fewer counts. This is noisier. And here's our event-by-event motion correction. The pancreas is one thing. Actually, the kidney's better. Sometimes I think God gave us the kidney as an in vivo phantom that we can look at resolution on, because if you look really, really carefully there, you can see how the little substructures of the kidney get blurred out. Here they're too noisy, and here we're pretty happy with it. No ground truth, no gold standard, but we're pretty happy with how things seem to be working out there. A couple other little issues, and there's a lot of these. There's 2 PhD theses and a couple master's theses in this project easily. I mentioned this fitting of data and the time activity curves and all of that. And this is some of that data, and we have to decide how do we analyze it. There's lots of these kinetic models that we use. This is an example of a data set. Here's a subject. We kept them in for 2 hours. We gave them a break, and then we let them continue. We got 4 hours of data. I mean, this is clinically impractical. But we tried to analyze that data. We analyzed it with a one-tissue compartment model. That's the solid line fit, and that's not very good. Okay, we did it with a 2-tissue model. That means we allow for a free and a bound compartment. Good fits there. Unfortunately, the numbers became, the more you ask for these models, the more numerically unstable they have. Then we tried some other intermediate models, something called MA1, or this is the simplified reference tissue model. I know that's being used there. And these models tend to describe the data. Maybe they're the Goldilocks methods. The problem is that they need a lot of data. That if we tried to limit it to an hour or an hour and a half of data, if we compare, one of the standard things we do is compare analyzing the first hour to 2 hours, etc. You hope that the numbers stay the same. That says you understand the system, and they're not. They're changing. So we have a lot more to learn. So we have a lot of questions. We have very few answers. We'd like to understand all the variability we're getting. There's a lot of variability between subjects. Maybe it's good variability. Maybe it's really reflecting variations in beta cell mass, or maybe it's noise, or metabolites. We'd like to optimize the scan duration, figure out how long we have to do. We have to figure out how to correct for this respiratory motion. It's theoretically possible. I know that the type 1 diabetics have the same amount of uptake, they just breathe differently. Because if we breathe more, it's blurring out the signal, it'll look smaller. We don't think that's the case, but you can't absolutely rule that out. We also want to decide when we analyze it. Our gold standard measurement is to use the input curve from an artery. And that's what those measurements analyses were. We took arterial blood samples to measure that. The arterial input function, not a great thing to do routinely. Okay? Alternatively, we'd like to use a reference region like the kidney or the spleen to be able to do that. Okay? And so we're trying to use that. And I mentioned the minus side, which we have problems with, but we're still trying to compare. Maybe there's metabolites in the kidney, but maybe they're similar in healthies and type 1 diabetics. We'll know that when we have that data collected. We'd like to get images. I've just showed you a few values. I'd like to get an image of this, like you saw with the brain. We saw a lot of brain images. We have a lot of counts, because there is a lot of reason why there should be regional variation. between the head of the pancreas and the body of the pancreas in the beta cells and how that changes. A lot of evidence that the loss of beta cells varies in different parts. And especially as we want to follow the natural etiology of type 2 diabetes, that'll be important. We'd like to know, do we need to do it the hard way with the model-based method, or this the simple imaging, standard uptake values? If you remember, I showed you the results where the standard uptake values were better. They were more significantly different. But bigger is not always better. It turns out there was a trick. One of the things that affects the amount you get in the system, of course, is how many receptors are there. But the other effects is the input, the delivery. It turns out that the plasma concentration of the type 1s at the same injected dose was lower. It cleared faster. So part of this difference is real differences, and part of this difference is that the tracer cleared faster. In other words, that the liver and the kidneys are behaving differently. So it looked good, but you were really measuring the wrong thing. No, our analogy for that would be when we go back to the brain. If we were studying alcoholism and somebody had a cirrhotic liver, their tracer might clear completely differently. We'd have much higher uptake in the brain, and we'd be measuring liver function with a brain scan. It's a very expensive way to do it. We're looking at how to tighten up our reproducibility. We'd like to follow people over time or follow people under drug treatment. And we need to understand, we've just begun to collect some of that longitudinal data to see how reliable this would be. These are all kind of the imaging questions and the biology questions. And then we have the harder questions. Is this target smart at all? Is this really going to be reflective? Are there better ones? We're also looking at GLP-1 receptors using extended analogs, ideally doing both of those in the same subjects, NIH permitting. And then will this really be effective, and will this help us with the natural etiology? So I've ended this section with more questions than I started with. We have a tool. It's not a knock-and-dead tool. It's interesting. It might help us develop a drug. It's not probably ever going to be individually diagnostic to say, but it might help us decide, learn a little bit more about the disease, and then learn a little bit about perhaps some drug treatments that might help. So let me end with the acknowledgments. It takes a village to do PET. That's my village. So there's about 50 people or so in our PET center in a wide variety, starting from graduate students and postdocs and statisticians and programmers and physicists and chemists and cyclotron engineers and clinicians and nurses and technologists, and as well as all of our collaborators in the various departments. I want to emphasize those people who I've shared their primary data in the talk today, our collaborators in different departments, of course, all of our sponsors for all the studies, and I thank you for your attention.
Pressure from outside to see specific avenues from pharmaceuticals, from clinicians, or maybe even from legal.
So, pressure. We certainly, if pressure comes with money, we're very happy about that. Sometimes our lead, you're looking where the opportunities are and where you have some basics of knowledge. I mean, it's challenging to come up with the right molecule that's going to be useful in the right target, that you're going to have the right population, that you're going to be able to interpret it. So I don't know if I would call that pressure, but certainly those are places where we're looking for the opportunities. When do you have the right situation? You have the right patient population available to you. You have a right tracer or a right possibility for tracer that's likely to be available. Best case, you have an animal model of that disease that you can understand it a little bit better, go back to the animal model to be able to tease that. It's rare you get that whole package together, and you can see that sometimes that we're battling that with looking at a different case. But there's issues where we're trying to say what's going to be useful diagnostically in individual patients. Will this be something, as we move forward and doing more and more into cancer, which I haven't showed you today, there's going to be lots of really great opportunities when we have very specific agents targeted to specific mechanisms that they may be very focused on one cancer type or one mutant type within a cancer to be able to do that. So the cool part is that we have a lot of possibilities, and the trick is basically to choose reasonably well, which is not so simple. So